Excellent. C stands for complementary. CDNA means complementary DNA. So these are the DNA that are synthesized from mRNA. So using mRNA as a template, we'll synthesize DNA. Yes. So now they're asking, what is the question? They want to know like with which enzyme we can synthesize cDNA from mRNA. You got the question? So we just want to find out the enzyme that helps in copying of cDNA or the synthesis of cDNA from mRNA. So we'll go through the options now. First option, restriction enzyme. So what's the function of restriction enzyme, students? Yes, you remember we use this enzyme to cut the DNA. Yes, so the function of restriction enzyme is to cut the DNA into fragments. So is this the enzyme we need now? No. So this is the wrong option. So we'll cancel it out. Next option, DNA polymerase. Come on, tell me the function of DNA polymerase. Very good. It's the enzyme we need for DNA replication, right? For adding deoxyribonucleotides to the growing chain. So again, this is not the enzyme. Here we are not synthesizing DNA from mRNA, but we are duplicating tRNA. So this enzyme is out of the question now. Next option, that is adenosine deaminase. Adenosine deaminase. Now this adenosine deaminase is also called adenosine aminohydrolase. So what is it called, students? This adenosine deaminase is also called as adenosine aminohydrolase. Now what's the function of this enzyme? Let me tell you. This enzyme is involved in purine metabolism. In purine metabolism. Students, you can take down this option and explanation for this option. Yes, so this adenosine deaminase or diaminase is also called as adenosine aminohydrolase, and the function is this enzyme is involved in purine metabolism, that is adenosine and guanosine metabolism. Clear? So again, this is not the enzyme we are looking for. So adenosine deaminase is out. So the remaining option is very good, reverse transcriptase. So it is this enzyme which helps us to produce cDNA from mRNA. So which is the enzyme, students? It is reverse transcriptase. Clear? Shall we move to the next question? Okay, next question. Which one of the following statement is true for protein synthesis? Your options. Option A, amino acids are directly recognized by mRNA. Option B, the third base of the codon is less specific. Option C, only one codon codes for an amino acid. Option D, every tRNA molecule has more than one amino acid binding site. So, let's read out the question again. Which one of the following statement is true for protein synthesis? So, among these four options, you have to find out the true statements regarding protein synthesis. So, we'll go from option D, that is from below. Yes? Every tRNA molecule has more than one amino acid attachment site. Now, go back and recall the tRNA structure. So, how many sites does it have for this amino acid binding? You all remember the structure? Yes? The structure of tRNA, chlor leaf structure. So what is the question here? It says that the tRNA, one tRNA molecule will have more than one amino acid binding site. Is it so? First of all, where do we find this amino acid binding site? Is it towards the 5' end or 3' end? Very good, students. It is towards the 3' end. So, one molecule of tRNA will have one amino acid binding site, not more than that. So, this option is wrong. Okay, let's go for the option C. Only one codon codes for an amino acid. Is it like all amino acids what we have are coded by only one amino acids? Very good, students. You know what is degeneracy, right? So again, this option is wrong because there are some amino acids which are coded by more than one codon. So can you give me some examples? In the last class, I mentioned one. In the last CET class. It was example for leucine. Yes. So for example, CUU, then CUA, CUG, and CUC. So all these codons, they code for one amino acid, that is leucine. So again, this option is wrong. So there are more than one codon which can code for amino acid. Now let's go for the option A. Amino acids are directly recognized by mRNA. Is it so, students? So whatever the amino acids that are there in the amino acid, that are there in the cytoplasm, will directly come and bind to this mRNA. Then what is the function of this tRNA? Why should a cell make tRNA? Yes, our amino acids which are present in the cytoplasm, they cannot directly recognize mRNA. So they need an interpreter. Yes, and that interpreter is tRNA. So this tRNA will get the amino acid to the protein synthesis site. So again, option A is wrong. So amino acids are not directly recognized by mRNA. So the correct answer is third base of the codon is less specific. So in this example, so it is because of the third member of the codon. You can see only the third member is changing in every codon, whereas the other two codons or the other two members, they remain as such. Correct? It's only the third member of the codon is changing. So we can say that the third base or the third member of the codon is less specific. So because of this third member, we have more than one codon coding for amino acid. Clear, students? So among all this statement, the true for the protein synthesis is the third base of the codon is less specific. Clear? Shall we move to the next question? Okay. Which antibiotic inhibits interaction between tRNA and mRNA during bacterial protein synthesis? Your options: erythromycin, streptomycin, neomycin, tetracycline. So let me read out the question. Which antibiotic inhibits interaction between tRNA and mRNA during bacterial protein synthesis? So before going to the options, first we'll know the meaning of antibiotic. So any one of you, not all, any one of you, stand up and tell me what is antibiotic or what antibiotic is. Sure, you would have come across this term when someone is suffering from a cough, yes, or some fever. The doctor will prescribe some antibiotics, correct? So what is antibiotic, students? Very good. These are the substances that destroy or inhibit the growth of microorganisms, especially bacteria. So what are antibiotics? These are substances, yes, that kill or inhibit, means they stop or they hinder the growth of bacteria.